Good morning friends, how are you? I hope you all are safe at home. Today in this session I am going to discuss few topics of chapter 2nd, that is sexual reproduction in flowering plant of 12th standard. Today's topics for discussion are adaptation promoting cross pollination in nature, that is also called as outbreeding devices, development of male gametophyte, growth of pollen tube, artificial hybridization. First of all, let me make you aware about the different outbreeding devices. So let us see. First outbreeding device is unisexuality. As you have learned that unisexual means both male and female parts of the flower are present in different flower or different plant. So if the flowers are unisexual, then only cross pollination can be achieved. For example, papaya and cucurbits. Here in the given figure, you may observe that in papaya, androecium, that is male part, and gynoecium, that is female part, are present in different flower. So in this case, only cross pollination is possible with the help of some pollinating agent. Second outbreeding device is dichogamy. It occurs in bisexual flower, where anther and stigma mature at different times. Due to this, self-pollination is inhibited and cross-pollination is preferred. Example of dichogamy is sunflower and salvia. Third outbreeding device is self-sterility. In this case, pollen grains of a flowers are unable to perform the process of fertilization due to its sterile nature, though they reach the stigma of the same flower, but due to the presence of sterile genes in the pollen grains, they are unable to fertilize the egg. Examples are potato, tobacco, crucifers, including Gramineae and Orchidaceae family. Next outbreeding device is heterostyly. In this case, flowers have different types of heights of styles and stamens. The stigma and stamen are not in the same level, and due to this, self-pollination is prevented and cross-pollination is achieved. Example, primose. Another outbreeding device is hercogamy. In some flower like Gloriosa and Calotropis, there are some physical barriers between male and female part, that is anther and stigma, and this prevents self-pollination and preferred cross-pollination. Last outbreeding device is self-incompatibility. It is the inability to produce functional male and female gametes for fertilization in the same plant. Incompatibility may be interspecific incompatibility, that is between two different species of same genus, or intraspecific incompatibility, that is within the same species. In the given figure, you may observe self-incompatibility. Next topic is development of male gametophyte. Pollen grain germination starts before it is shed from the anther. During this process, what is happening? Nucleus of the pollen grains divides mitotically to form two nuclei, that is a vegetative or tube nucleus and a smaller generative nucleus, and thereby forming two cells, vegetative cells and generative cells. In this case, vegetative cells are larger and generative cells are smaller in size. in most of the cases at this stage, means at this two-celled stage, the pollen grains are transferred to the stigma. But in some cases, what is happening, like in some cereals, the generative cells further divide mitotically to form two male gametes before pollination. And at this stage, that is at three-celled stage, this pollen grain reaches to the stigma. In the given figure, you may observe the vegetative cell and generative cell of a pollen grain. Let us see about growth of pollen tube. As soon as the pollen grain reaches the sticky stigma, it absorbs its secretion and water. As a result, it swells up. Due to this, there is rupture at one end of the germ pore, as a result of which the thin intine comes out in the form of a pollen tube, which moves towards the styler region due to chemotropism, that is movement towards the chemical present in the style. Here the generative cells form two male gametes after mitotic division. The pollen tube enters the ovule through a small opening, and that small opening is called micropyle. Inside the embryo sac, the tip of pollen tubes rupture and the two male gametes are released into the egg apparatus. Inside the embryo sac, one male gamete fuses with egg nucleus to form zygote, and which is called as syngamy or true fertilization. And other male gamete fuses with polar nuclei called as triple fusion to form pen, that is primary endosperm nucleus. And this primary endosperm nucleus provide nutrition to the developing embryo. Since fertilization is twice, so it is called as double fertilization. Here you can observe the LS of flower showing growth of pollen tube. Last topic of today's session is artificial hybridization. It involves two processes, that is emasculation and bagging. So let us see what are these two processes. In emasculation, there is removal of male part, that is anther, from the flower bud before the anther dehisces. Now the question arises, why it is done? It is done to get desirable or useful character during hybridization or crossbreeding. After that, desired pollen grains are dusted over the emasculated flower and it is covered with a bag of suitable size so that it prevents the contamination of stigma with unwanted pollen, and this process of covering is called as bagging. Generally, butter paper is used to cover the flower. So this is all about today's session. Thank you and have a nice day.